Welcome all to today's guidance lecture on pharmacognosy and phytochemistry second, as well as herbal drug technology. In this section, we learn about different multiple choice questions, some key concept. So in this previously lecture, we learn about pharmacognosy 1. In that pharmacognosy, the word derived from Greek words, that is pharmakon means drug and gymnosco means to acquire the knowledge. So in that subject, we learn about metabolic pathways in higher plants and their determination, brief study about basic metabolic pathways and formation of different secondary metabolites. So this bit will questions asked many last examinations. Basic metabolic pathways. So I have to especially ischemic acid pathway, amino acid pathway. So I have to study in this section. So metabolic pathway first in higher plants involve biochemical process, metabolism, conversion of one chemical form into another chemical form with the help of enzyme. Enzyme it acts as a catalyst. Metabolism have no. So this required for support the growth, development and reproduction. This pathway are divided into primary metabolism and secondary metabolism. Primary metabolism essential for photosynthesis, respiration, nitrogen and carbon utilization, lipid metabolism, and secondary metabolism it required for defense signaling and ecological adaptation, including alkaloids, terpenoids, and phenolic compounds. Next important that is ischemic acid pathways. These involve synthesis of aromatic amino acids. So amino acids that is heard about in biochemistry subject, that is essential amino acids and non-essential amino acids. Essential amino acids are not synthesized in body; they need to administered. That is have heard about essential amino acids, that is tryptophan, alanine, phenylalanine, methionine, sulfur-containing amino acids. So these are the amino acids: phenylalanine, tryptophan, tyrosine, and secondary metabolites like alkaloids. Alkaloids are nitrogen-containing compounds that is basic in nature. Flavonoids, that is antioxidant properties. Plant defense and environmental interaction. These are next acetate pathways. Use acetyl-CoA produce fatty acid. Amino acid pathway, amino acid precursor for secondary metabolite like alkaloid, terpenoids, phenolics, which are vital for plant growth and defense. Next, radioactive isotope in biogenetic studies. So radioactive isotopes are essential. Radioactive isotopes are essential tools for investigating plant metabolism, especially in biogenetic studies. They help elucidate biosynthetic routes, track metabolite formation, and determine rate of biosynthesis in plant metabolism. Next, turn to the multiple choice questions. First question, that is, which of the following secondary metabolites is formed through ischemic acid pathway? A. Terpenoids, B. Alkaloids, and C. Tryptophan, and D. Polyketides. So which of the following secondary metabolite is formed through ischemic acid pathway? That is tryptophan. C is the correct answer. Next, which of the following carminative? Option A. Coriander, B. Fennel, C. Cardamom, D. All. So coriander, then you have heard about fennel, badishep, cardamom, elaichi. So these are all are carminative. So answer D is the correct. Next question, turn to the which of the following secondary metabolite is derived via the acetate pathway? Option A. Flavonoids, B. Steroids, C. Phenolics, and D. Anthraquinones. So D is the correct answer. Anthraquinones. Next, radioactive isotopes are used in biogenetic studies too. A. Study enzyme kinetics. B. Study tool for investigating plant metabolism, especially in biogenetic studies. C. Measure pH changes, determine protein structure. So B. Tool for investigating plant metabolism, especially in biogenetic studies. So B is the correct answer. Next, unit 2, in that general introduction. Alkaloids, question will ask in alkaloid point. In that vinca, so vinca, it active constituent, that is vincristine and vinblastine, used in the cancer treatment. Rauwolfia, Rauwolfia contained reserpine, it is used in hypertension, antihypertensive agent. Also anxiety, belladonna, atropa belladonna, parasympatholytic drugs. Opium, pain relief, phenylpropanoids and flavonoids, antioxidant. Steroid, cardiac oxide, that is cardiotonics, cardiac oxide, digoxin and digitoxin, you have heard about Propenthus, Arjuna. These are the cardiotonics. Triterpenoids, licorice, anti-ulcer drug, Diascora, digitalis, anti-cardiotonics. Volatile oils in the mentha, clove, lavang, I will me. Clove, that is eugenol, active constituents, cinnamon, and the fennel and coriander. Tannins, catechub, petrocarpus, resins, benzoin, gagul, ginger, asafoetida, myrrh, colophony. Glycoside, senna, aloes, bitter almond. So alkaloids, these are the nitrogen containing compound that are biologically active and exhibit therapeutic properties. Example, that is vinca, already told, vincristin active constituent that is used in cancer treatment. Raudfia contain reserpine, treat hypertension. Belladonna, parasympathetic, opium, pain relief. Phenylpropanide and flavonoids, that is anti-inflammatory and antioxidant, prevent the inflammation. Lignins found in seeds used as antioxidant. Tea contain catechin, antioxidant. Ruta contain rutin, blood vessel and inflammation prevention. Steroid and cardiac glycoside, triterpenoids, in that licorice contain glycyrrhizin, used in anti-inflammatory and anti-ulcer effect. Diascora contain dioxygen, used in synthesis of steroids. Digitalis contain digoxin, used for congestive heart failure treatment. Extraction through solvent extraction. Next, volatile oils. Volatile oils, these are odorous aromatic compounds used in perfuming, flavoring, and medicinal purposes, as well as cosmetics. The cosmetics means that is articles intended to utilize for beautification purpose. Volatile oils. So in that, mentha contains menthol, used for cooling agent. Clove, eugenol, used as analgesic. Cinnamon contains cinnamaldehyde, used for carminative. Fennel contains anthol, used for carminative. Eucalyptus, antiseptic. So this isolation techniques of also volatile oils, or these are the essential oils also. So this isolation of volatile oils, steam distillation, have to remember steam distillation techniques. Also hydrodistillation, inflorescence, E. coli method. Another different have to peppermint have volatile oils, sandalwood, cinnamon, lemongrass, carve, anise, etc. So you have to remember tannins. Tannins are polyphenolic compound with astringent properties and used various medicinal applications. Example, catechol contain catechins and used as a astringent. Petrocarpus used for wound healing and as a astringent. Next, resins. Resins are amorphous plant product used for their aromatic and medicinal properties. Benzene used in externally as antiseptic, protective. Gagul, cholesterol reduction is hypolipidemic, decrease the cholesterol level. LDL, VLDL, low density lipoproteins, very low density lipoproteins, and high density lipoproteins. You have heard about. So this Gogul decrease the cholesterol level. Asafoetida used in digestive aid. Myrrh, anti-inflammatory, antimicrobial properties. Colophony. Glycosides. Glycosides, these are organic compounds of plant and animal sources consisting of sugar and non-sugar, glycon and glycon parts, components that have medicinal properties. Senna contains sinucide used as a laxative. Aloes contain aloin used as a laxative and skin care. Bitter almond contain amycadlin, digitoxin as a cardiotonics. Next, iridoid and terpenoids in that gentine contain gentle peroxide used for digestive disorder. Artemisa contain artemisinin used for treat malaria. Taxus contain paclitaxel used in cancer treatment. Carotenoids uh, in Marathi that is gajar, found in fruits and vegetables, used for vision and antioxidant benefit of carotenoids. Next, we turn to the multiple choice questions. First question, that is, which alkaloid is the primary active compounds in Rauwolfia serpentina? Option A, that is morphine, B, reserpine, C, atropine, and D, codeine. So reserpine, Rauwolfia serpentina active constituent, that is B, reserpine. So answer is B is the correct. Next, which of the following is a not identification test of alkaloids? Option A, Mears reagent, B, Hanger's reagent, C, Wagner's reagent, and Rangendorf reagent. And option D, killer test. Which of the following not identification test of alkaloids? So D is the not killer test, that is identification test, that is glycosides. Next, which of the following example of propane alkaloids? A, belladonna, B, coca, C, vidania, and D, all of the above. So all of the above, that is propane alkaloids. Next, the lignins from ruta exhibit which primary activity? A, antiviral, B, antioxidant, C, antidiabetics, D, antimicrobial. So lignins, antioxidant means these are the substances which prevent the oxidations. Next, which of the following stages of life cycle of ergot? A, overwintering stage, B, stage of sexual reproduction, C, stage of asexual reproduction, and D, all of the above. So all of the above is the correct answer. D is the correct answer. Next, which of the following identifies test of cardiac glycoside? So previously question will discuss which of the not identification test of alkaloids. Here question asks which of the following identification test for cardiac glycoside? A, synostotics, B, killer test, C, foaming test, and D, all of the above. So killer test is a identification test of cardiac glycoside. Synostotic test, that is flavonoid test identification. Foaming test, saponin glycoside identification test. You have to also remember this. Next, the major component of clove oil: A, menthol, B, eugenol, C, lanolol, and C, cinnalol. So eugenol is the major active components of clove. B is the correct answer. Next, which of the following volatile oils drugs? A, cardamom, B, caraway, C, fennel, and D, all of the above. So D is the correct answer. Next, primary tannin present in catechol: A, gallic acid, B, catechin, C, ellagic acid, and D, tannic acid. So B is the correct answer, catechin. Next, which plant is commonly used as a source of tannins for healthier leather tanning? A, Petrocarpus, B, Terminilla chibula, C, Acacia catechu, and D, Quercus infectoria. So Acacia catechu is the correct answer, C. Next, the resin obtained from gogul is primarily used for A, antihypertensive activity, B, hypolipidemic effect, C, antidiabetic properties, and D, antipyretic effect. So B is the correct answer, hypolipidemic, decrease the cholesterol level. Next, the primary active glycosides in senna leaves responsible for its laxative effect: A, anthraquinone glycoside, B, cardiac glycoside, flavonoid glycoside, phenolic glycoside. So A is the correct answer, anthraquinone glycoside. Next, third unit we learn isolation and identifications and analysis of phytoconstituent, terpenoids, menthol, citral, artemisinin, glycoside, alkaloids, resins. First, that is, which of the following terpenoids is the main constituent of peppermint oil? A, menthol, B, citral, artemisinin, and camphor. So A is the correct answer, menthol. Citral K terpenoids is widely used in the production of A, anti-malarial drugs, B, vitamin A, C, fragrance and lemon flavoring, D, steroids. So fragrance and lemon flavoring. Next, artemisinin is the primary isolated from which plant? A, Artemisia abacidum, Artemisia anna, Artemisia vulgaris, and Artemisia ranculus. So B is the correct answer. Next, question number 4. Glyceric acid is derived from A, Glycyrrhiza glabra, Senna alexandra, aloe vera, digitalis purpura. So A is the correct answer. Rutin glycoside is primarily recognized for its A, antipyretics activity, B, antioxidants. So rutin is antioxidant. B is the correct answer. Next, which alkaloid is obtained from bark of Cinchona pescis? A. Reserpine, B. Caffeine, C. Quinine, and D. Atropine. Quinine, antimalarial, and quinidine, that is antiarrhythmic. I also remember. Next, digitized leaves are adulterated. Which of the following? A. Primrose leaves, B. comfrey leaves, C. leaves of Fabaceae thapsus, and D. all of the above. So D. all of the above. Next, caffeine, a purine alkaloid, is primarily found in A. coffee and tea leaves, B. coca and cola nuts, C. both A and B, D. only tea leaves. So answer C. both A and B. Coffee and tea leaves, coca and cola nuts. Next, reserpine is an indole alkaloid, is isolated from Rauwolfia serpentina. So A is the correct answer. Next, podophile toxin used in anti-cancer therapy is extracted from Podophyllum hexadrum. Next, which of the following biological source of alloy? A. alloy barbadis Miller, alloy Baker, alloy Ferox Miller, alloy African Miller, D. all. So answer D is the correct answer. Next, which techniques is most commonly isolation of volatile terpenoids like menthol? A. steam distillation, B. succinate extraction, C. maceration, and D. sublimation. So steam distillation is the correct answer. Forskolin is isolated from which medicinal plant? Cyanosides, known from their laxative effect, are primarily extracted from Cassia angustifolia. Which of the following is a characteristic functional group in artemisinin? Peroxide bridge. Caffeine, a central nervous system stimulant, is mainly extracted from caffeine CNS stimulant. All of the above. Next, which method is used for extraction of phytochemicals that are succinate extraction, microwave assisted extraction, a steam distillation, and all of the above? So all of the above is the correct answer. HPLC separate compounds primarily based on A. molecular size, B. polarity, B. boiling point, and D. electrical charge. Polarity is the correct answer. Next, which of the following techniques is most suitable for separation of volatile compounds? Gas chromatography. Next, infrared IR spectroscopy is primarily used to identify A. functional group of molecule, B. molecular mass, C. elemental compositions, and D. optical isomer. So functional group incompatibility determination IR spectroscopy. So also remember answer A is the correct. Next, electrophoresis separate by a molecule based on their molecular weight and solubility. Polarity and volatility separate DNA, RNA, protein molecule based on their size and electrical charge. D. adsorption and affinity. So answer C is the correct based upon charge and size. Next, which chromatography techniques involve the use of solid stationary phase and liquid mobile phase? Gas chromatography, column chromatography, paper chromatography, and ion exchange chromatography. Water purification, ion exchange chromatography utilized. Column chromatography is the correct answer, B. Next, which detector is most commonly used in gas chromatography? GC for organic compounds. What is the principle of thin layer chromatography? TLC. Mixture compounds are separated between stationary phase and liquid mobile phase by different affinity between two phases. Next, which analytical techniques is best determine molecular weight of phytochemical? That is mass spectroscopy, UV visible spectroscopy, nuclear magnetic resonance NMR spectroscopy, and gas chromatography. So mass spectroscopy determines molecular weight, UV visible spectroscopy determine saturated and unsaturated compounds, impurities. That is the role of UV spectroscopy, sigma to sigma transition, pi to pi transitions. So energy will absorb and excite the molecule. You have heard about nuclear magnetic resonance determine the signals. So A is the correct answer. You have to remember. Next, we'll turn to the herbal drug technology. Herbs as raw material. A definition of herb: herbal medicine. Herbal medicine product, herbal drugs, preparation source of herb selection, identification and authentications of herbal materials, processing of herbal raw material. So identification and authentications is a very important of herbal drugs. Means that is study of crude drugs, official titles, synonyms, biological origin, geographical distribution and history, cultivation and collection, plant protection, macroscopic and microscopic study, chemical constituent means active constituent, chemical test means identification test also, therapeutic and pharmaceutical uses, storage, adulterant. So these are the required for a study of pharmacognostic drugs. Indian system of medicine, basic principle involved in Ayurveda, Siddha, Unani, and homeopathy. Preparation and standardization of Ayurvedic formulation. Standardization means standard. So herb and herbal medicine, use of plant or plant extract for medicinal purpose. Herbs are plants or plant parts such as leaves, flowers, seeds or roots used for medicinal or cooking purpose. In herbal medicine, these are the herbs often prepared as tincture, capsule powder or topical applications. Herbal medicine refers to the use of this plant or plant-based substance to treat the health conditions. This type of medicine can be considered in part of traditional or alternative medicine remedies such as tincture concentration herbal extract typically made up with alcohol or vinegar, capsule tablet containing powder or concentrated plant material, topical creams or oil, ginger for nausea or digestive issues. Basic principle involved in Ayurveda, Siddha, Unani and homeopathy treatments. Homeopathy, like your likes, homeopathy, allopathics treat symptoms, Unani, uh, humus, Siddha based on the balancing of body internal energy similar to the Ayurveda. Achievement have discussed in previous. Next, preparation and standardization of Ayurvedic formulation, Arista and Asva. So question will ask which alcohol generate the formulations, self-generate. So are Aristas and Asvas fermented liquid formulations with herbs and other substances. Standardization ensuring proper fermentation, potency, quality control, alcohol content and consistency. So standardization means standard to find out the standard of that formulations. Gutika, solid tablet made from powder herbs. Churna, powder herbal formulation. Leha, semi-solid herbal paste mixed with honey, ghee or sugar. Basma, calcinated heated metal or mineral preparation in the form of fine ash. Next, which of the following used as herb? A plant used for medicinal purpose only. B, a plant that is used for flavoring food. C, traditional medicine. D, all of the above. So D, all of the above. Next question, what is herbal medicine? A, use of synthetic chemical to treat disease. The use of plant-based substance for therapeutic purpose. C, the use of animal product for cure disease. The use of water-based solution for medicinal treatment. So herbal, plant-based substance. B is the correct answer. Next, which of the following is the essential part of selection and identification process of herbal materials? A, random selection without any standardization. B, identification of plant species including botanical name. No need to authenticate origin of plant. D, using only synthetic methods of identification. So B is the correct. Identification of plant species including botanical name. Next, question number 4. The processing of herbal raw materials include which of the following steps? A. Harvesting and drying. B. Purification and extraction of active constituent. C. Standardization for quality control. D. All of the above. Next, which of the following is the basic principle of Ayurveda? Question is that which of the following basic principle of Ayurveda? A. The balance of 5 elements and 3 dosas. The use of only synthetic chemical. C. The importance of fasting and detoxing. Emphasis on mental health but not on physical health. A is the correct answer. Balancing 5 elements and 3 dosas, Vata, Pitta and Kapha. Next, which of the preparation is typically used in Ayurveda medicine as an alcoholic extract herb? A. Gutika, B. Aristan Yasva, C. Basma and D. Churna. So this question will ask in GPAT examinations. Aristan Yasvas. Next, what is the Basma in Ayurvedic medicine? A. Tablet form of herbal medicine. B. A fine powder preparation obtained by incinerating herbs. C. An alcoholic extract. D. A syrup made from herbs. Next, the role of pharmacognosy in allopathy and traditional system of medicine involves A. Identifying toxic plants. B. Understanding medicinal properties of plant-derived compounds. C. Creating synthetic substituents for herbal remedies. D. All of the above. So B is the correct answer. Next, in the preparation of Ayurvedic formulation, what is churna? A liquid preparation. B. A solid tablet. C. A powdered form of herbs. And D. A syrup. A powdered form of herbs. Next, what is standardization of herbal formulation ensure? Standardization is standard. The formulation has no side effect. The formulation contains consistent amount of active ingredient. So B is the correct answer. Next, what does the Gutika refers to Ayurvedic medicine? A. Tablet pills from the herbal extract. B. Alcoholic preparation. C. Powder mixture. Gutika, that is tablet or pills from herbal extract. Nutraceutical. Next. First question, which of the following true points regarding nutraceuticals? Nutraceuticals are products derived from food sources that provide additional health benefit beyond the basic nutrition. Also, B, that is this can include like vitamins, minerals, amino acids, fatty acids and herbs, fish oil, omega-3 fatty acid, turmeric, curcumin, green tea extract, garlic, which are taken to support overall health, prevent disease or treat specific health condition. C, the term nutraceutical is a combination of two words, nutrition and pharmaceutical. So D is the all of the above. D is the correct answer. Next, which herb is used for reduce nausea? A. Ginger, B. Ginseng, C. Amla, and D. Garlic. So ginger. Next, which following herbs is known for its role in boosting the immune system and has antioxidant properties? Amla, antioxidant properties. Next, which of the following is used as a sweetening agent? A. Ginseng, B. Garlic, C. Honey, and D. Amla. Honey is a sweetening agent. Herbal cosmetics. So cosmetics used for beautification purpose. Which of the following is commonly used as raw materials in herbal cosmetics for its antioxidant properties? A. Aloe vera, B. Gums, C. Vitamin C, and D. Honey. Vitamin C. Next, what is the role of herbal excipient in formulation? A. To improve the taste of the formulation. To give the formulation desired texture and consistency. Both A and B. And D, none of this. So C is the correct answer. Next, which of the following is used as a natural colorant in herbal cosmetics? A. Ginseng, B. Beetroot powder, C. Curcuma, and D. Fenugreek. So beetroot powder. Next, phytochemistry, a novel dosage form, are primarily used to A. Enhance the bioavailability of herbal ingredient, B. Improving the taste of herbal formulation. C. Extend the shelf life of herbal drugs. D. Reduce the cost of herbal medicine. A. Answer is the correct. Next, which organization provide guidelines for the evaluation and stability testing of herbal drugs? A. WHO. B. FDA. C. Both A and B. D. None of this. So answer is WHO, World Health Organization. Next, what is does IPR stand for in the natural product? A. International Product Regulation. B. Intellectual Property Rights. Integrated Plant Resources. And D. International Patenting Requirement. So IPR stand for means Intellectual Property Right. IPR. But here question that is IPR stand for in the context of natural product. Yeah, once I have to view this answer or correct it. So IPR generally IPR means intellectual property right. Next, which act regulate the manufacture of ASU Ayurvedic Siddha Unani drug in India? A. Narcotic Drug and Psychotropic Substance Act 1985. B. Drug and Cosmetic Act 1940. C. Drug and Magic Remedies Act 1954. D. All. Drug and Cosmetic Act 1940. That is the correct answer. Next, which of the following best describe the current scope of herbal drug industry? A. Limited traditional medicine only. B. Expanding the do grow in consumer preference for natural products. C. Focuses slowly on synthetic drugs. D. Only applicable for local use, not global market. So B is expanding growing consumer preference for natural products. Next, which of the following Indian institutions involved in the research and development of medicinal and aromatic plants? A. Indian Institute of Integrative Medicinal Medicine, Council of Scientific and Industrial Research (CSIR), Central Institute of Medicinal and Aromatic Plants (CIAMP). So D, all of the above. D is the correct answer. Next, Schedule T of Drug and Cosmetic Act 1940 focus on which of the following? Good manufacturing practice, Ayurvedic Siddha and Unani. So B is the correct answer. Next, future prospect of herbal drug industry primarily depends on A. Availability of synthetic alternative. B. Regulatory restriction plant-based product. C. Consumer demand and natural and organic product. D. Lack of technology advancement. So C is the correct answer. Consumer demand for natural and organic product. Next, fifth, primary benefit of implementing Schedule T in the herbal drug manufacturing is A. Ensuring standardization of quality and safety of herbal product. B. Reduction in the cost of production. C. Limiting the scope of herbal medicine uses for rural areas. D. Increasing reliance on synthetic chemicals. A is the correct answer. Next, which of the following Indian institution involved in research and development of medicinal and aromatic plants? So, Indian Council of Medicinal Research (ICMR), Council of Scientific and Industrial Research (CSIR), and also NIPER. D, all of the above. Next, previous year question papers questions. Explain method of collection of barks. Write general test for alkaloids. Write general and chemical test for tannins. Pharmacognosy and phytochemistry question paper. This is what are the proteins and enzymes. Enlist the types of vascular bundles. Explain therapeutic uses of papain. Write a note on auxins. A 10-mark question. What is polyploidy, mutation, and hybridization? Explain their application in detail. Define and classify alkaloids. Explain method of extraction and general chemical test for alkaloids. Explain in detail general extraction lipids. Define and classify volatile oil. Enlist properties of and application of volatile oils. Next, 5-mark question. Write biological source, chemical constituents, and applications of honey. Add a note on preparation of honey. explain natural allergens. Next, give nutritional requirement, growth, maintenance of plant tissue culture. Draw well-labeled anatomical diagram of leaf. Add a note on mutation and give its significance. Classify regions and detail along with example. Explain on leaf constituents in detail. Write a note on uh, cotton and jute. Explain flavonoids and biological importance. Write chemical test for gentine and triacanth. So this is a those are preparing for university examinations. This question paper. Next, herbal drug technology questions, that is, write a note on homeopathic system of medicine. Explain detail possible side effect of interaction of garlic. Brief note on herbal origin, perfumes. Define natural binders along with classification and advantages. Write a note on natural sweeteners. Define patent, trademark, and bioprivacy. Write a note on ashwagandha nutraceutical. 10 marks. Explain good agriculture, cultivation, and medicinal plants. Define nutraceuticals. Explain in detail WHO ICH guidelines for assessment of herbal drugs. So in this lecture, you have to remember this point. In GPAT point of view or any competitive examinations, questions will highlight it, that is chemic acid pathways, acetate pathway, another. So this is uh, very important. Alkaloids. So alkaloid in that indole alkaloids, vinca, phyostigma, naxomica, rauwolfia, ergot. And next, imidazole, pilocarpine, that is used in treatment of glaucoma. Tropane alkaloids, that is stramonium, devil's apples or thorn apples synonym. Belladonna, anticholinergic drugs, hyoscine, datura, coca, local anesthetics agent. So these questions will ask me synonyms, biological source, chemical constituent. Make it the chart. You have to prepare the charts. You have to remember in cognosy subject. That is, make it the chart is needed. So these questions will ask means identification test, biological source, family. So this unit 2 is very important. You have to remember glycoside identification test, flavonoids identification test, isolation techniques, what Gutica, also Ashwa Aristas. So these all points are very important. Hope this lecture found to the helpful for your competitive examinations. Wish all the best for your examinations. Prepare a best. Definitely result will come a best. Thank you for listening and this watching this lecture. Thank you.